is that an interest that I have, a very strong interest, is making things that we learn in the basic research to make it apply to actual problems of human health. I'm going to discuss the capability that we now have through this bioinformatics approach to shotgun sequence to be able to make accurate, very rapid identification with information that can be used right away to treat a patient or to decide on safety of a food product or the safety of water that's being treated for human drinking source. Actually, very quickly, because I did not come to this sort of yesterday, but in fact, my whole career sort of has come to this point where we can take the kinds of information that we learned very early on. Very early on, back about 50 years ago, seems like a long time ago, all we could do was essentially study the phenotype, that is, culture things, determine whether they fermented sucrose or lactose, and did other kinds of things, and then we coded the data, developed the first program back in 1962 to analyze taxonomic data. We also moved from there to some of the work in the field, because I am a microbial ecologist, knowing that there were a lot of bacteria out there that we could not grow. But we developed a technique that allowed us to actually see these and published a paper on the non-cultural bacteria, which was very controversial at the time, but we discovered it because when we actually did culture and enumerated and then did fluorescent antibody, we were missing a lot of bacteria. We did microscopy to show that yes, this is a dormant stage. As I say, it was very controversial, and so since then, around 400 papers have been published and about 1,000 papers describing dormancy in gram-negative bacteria and related microorganisms. So these are two important points. The fact that we needed to find a way to use the genomics because the phenotype is not very precise. And the fact that we needed to identify everything that was in a sample, whether we could culture it or not, was really critical. Now, I've focused most of my work, as you know, on cholera, and in working in Bangladesh since 1975. We did make a discovery that the vector, and I am going to be very bold now and call the copepod a vector for cholera, because the copepods can carry up to about 50,000 colony-forming units, probably more Vibrio cholerae, and if ingested, particularly during a time of copepod abundance in the spring and fall, you get enough of the infectious dose to have enough cases of cholera. We, again, because I'm a very pragmatic person, we found a quick and easy solution to prevent cholera simply by filtering the water very crudely using Sericot, and that allowed us to reduce cholera by 50%. So I am keenly interested in the application. Been working on the genomics a long time. That's really a long time ago. Back in the 60s when we began to do base compositions and do hybridizations. And now we have the capability of next generation sequencing. That is what I find really exciting, and that's what we'll spend the rest of the time talking about. It was 16 years ago, and I know Claire Fraser's in the audience, Claire and I, the gang at Tiger, sequenced the first Vibrio cholerae in the demonstration that it had 2 chromosomes. I understand from talking with Dr. Qiagen from the postdoc who has his own company at booth here, that there's just been discovered a Vibrio with 3 chromosomes, maybe a megaplasmid, but the claim is 3 chromosomes, so that's in the last week or 2. So you have to, with Vibrios, you have to be pretty careful because that organism, about 80% of its genes can be laterally transferred. And here you can see the genomic islands that can enter and leave in the case of the small chromosome to the right, chromosome 2, and the large chromosome to the left. So it's a highly versatile organism. Now, what we have learned about Vibrios and the next generation sequencing approach is that what the factors that we apply can be employed in clinical microbiology across the board. It allows us to determine the presence of a variety of genes and functions, and that's what I will go into in a little bit more detail. So, setting aside on the left-hand side part of the slide here, these are all culture-based, targeted, single isolate detection to go to the universal detection of metagenomics and using algorithms that are really, really powerful and rapid. And that's what led me to form a company. I'm going to be very blunt and say that it's a lot easier to get money to do the work you really want to do by forming a company than it is to go to NIH for a run, right? So in that case, forming Cosmos ID in 2008 has been a very fortuitous and very, very exciting time. The metagenomics, for this crowd, I don't have to go into the details, but what we've been able to gather is a database of about 65,000 genomes, bacteria, viruses, fungus, and parasites. I want to make clear that with the algorithms that we've developed, the system that we've developed, we can identify at one time from the shotgun sequence all of the microorganisms. And we can give a proportionality of the representation as well as selectively identify genes that they carry. And this is all done literally within minutes. And the platform is agnostic. It can be on any of the commercially available sequencing machines, as you can see, Illumina, Thermo Fisher, PacBio, Oxford Nanopore. And we also spent a lot of time to provide a very easy human-computer interface so that the operation, you don't have to be a computer expert, you don't have to be a bioinformaticist. You simply enter your shotgun sequence and the kind of analysis that I'll be describing, you can get literally within an hour and a half between what has been developed at Qiagen, functioning as an end-to-end complete solution. So, GenBook is the database that we very highly curated to eliminate error and contamination. As we all know, we have introduced contamination either from, let's say, the human sample, eliminate the human sequences, or from the technician who's preparing the DNA, sometimes he introduces a contaminant. Or, let's be honest, even the reagents that are used for extraction can contain a contaminant. So we are able to curate and clean beautifully the DNA sequences for which go into the database. And we have more than 65,000 genomes, and we're entering genomes really on a daily basis, so it's constantly expanding. It is a system as well that I want to make sure I'm clear on that. It's the database, the algorithms, and the interface that allows a complete capability within minutes to be operated. These are just some of the examples of some of the work that we have underway. In fact, we have about 6 or 7 posters here at this meeting of various examples of heart valve, endocarditis identifications, the use of our technique, our system, to determine the improvement of reused water, Orange County water system, and with whom we have done some work, been able to show that there's a reduction after the filtration steps of chlorination of the pathogens, and it's a very nice, pleasant surprise, a reduction as well in the antibiotic resistance genes, so that the fear that there would be a transfer of antibiotic resistance to the natural background water flora does not seem to occur. And that, I think, is very important. So let me give you a full case history that is exemplary of what we can do. This is a study done in Calcutta in collaboration with the National Institute of Cholera and Enteric Diseases in Calcutta. we had sets of samples brought to us at phase 1. We looked at samples that they had done a whole series of lab tests, 26 laboratory tests, culture, using viral tissue culture detection and microscopic measurement of parasites, the detection of presence of parasites. So in the first lot they sent us, they had identified in the laboratory the pathogen, mostly Vibrio cholerae, which is all they were looking for. And then they sent us another set of samples where they couldn't identify what was there, but the patient really had very serious diarrhea and otherwise was considered to be a cholera patient. And then they sent us another one where they knew what was the pathogen, another set of samples they didn't know what the pathogen was. And in both phase 2 and 3, we also got samples that they tested for the 26 laboratory tests that served as controls. These were asymptomatic, either family members without cholera or diarrheal disease of any sort, or members of the community. So that they served as our controls, if you will. Now, this is the study then, 65 fecal samples, 17 were patients with what they could identify as they call the causative agent in Calcutta at the laboratory there. And then 28 patients where they knew they had diarrheal disease, but they couldn't really identify any one pathogen or any pathogen at all, and then the 20 healthy individuals. And so this way, and then the other thing we did was to download from the NIH Human Microbiome Project the healthy Western gut flora samples so that we can compare them with the healthy Indian population gut flora. This was very interesting because the results showed that, as you can see, the healthy control on the bottom left is the Indian population, and the healthy control, the NIH data set, is the bottom right. And then at the top, the known etiology at the left is where the Calcutta lab could identify the agents, but on the top right, they could not identify. And it turns out that a couple of things I think are worth pointing to you. The bottom left, healthy Indian metagenome, you can see is quite different and characteristic compared to the Western NIH database, Human Microbiome Program data, metagenomic data. And I think the gamma proteobacteria are the pink, and they're where most of the enteric pathogens are located, the Firmicutes. And then the Bacteroides entities, these are the green, and as you can see, in the bottom right, in the Western healthy individuals, mostly the Bacteroides are related, what you would say, the probiotic type microorganisms. And in the bottom left, you can see that that group here of the probiotic, and probably, this is a guess, it's still going to be looking into, maybe because they are more abundant. And as you may know, the Indian diet is almost every meal is yogurt or some sort of probiotic type foodstuff. And that may in fact be protecting the pathogens that are in the healthy Indian population gut. Now, by being able to identify the pathogens very specifically, it's useful to point out that this lot here is the Human Microbiome Program, the NIH data set, and this is the unknown etiology, and we're able to identify particularly this cohort, this set of microorganisms are mainly enterics. And so the unknowns were mainly the Enterobacteriaceae members, and we were able to pick it up when we could not isolate them or identify them on the traditional laboratory tests. And that the general mixture of the known and unknown sample set of various pathogens, which are all listed on the right-hand side. So we're able then to readily identify these pathogens, many pathogens. And this is another point that I would like to emphasize. The assumption we've made from Koch's postulates that there is a pathogen. We are finding that it's between 4 and 10 pathogens that are involved in the hospital-admitted patients with severe diarrhea. So I think it is time for us to rethink in the 21st century the Koch postulate approach, which worked very well for us for 100 years, but now we have very powerful tools, and we need to understand the interactions amongst pathogens that create then the severe disease state that requires admittance to the hospital. So that's a second important point from our studies. Now, from the known etiology set, we identified the genomes. That is, we characterized and identified the species, and this method of ours, this algorithm set, allows us to identify to species and strain. It is much more accurate, well, it is accurate compared to 16S. So that's an important point to make. Secondly, we also are able to identify specific genes, and that allows us to point out that in the known etiology of the typical enteric pathogens, plus we're able to describe what genes they carry to create the pathogenic state. And then for the unknown etiology, they're mostly in the Enterobacteriaceae, the enteric E. coli family, which includes the E. coli Shigella. And again, we can identify specifically the species and then as well the genes that are present that contribute to their pathogenic properties or state. And then in the unknown etiology, where predominantly we picked up Shigella flexneri, which was not able to be isolated and identified by a classical test in the laboratory in Calcutta. It's a very good laboratory, so I'm not implying that they are inefficient. They are very, very good. It's just that the laboratory tests are not effective and efficient compared to the metagenomic approach that we're providing. And so you can see that through another presentation, this is the human microbiome data set of Westerners, and then the control from the Indian population are rather distributed, have a greater diversity associated with them, and then the known etiologic agent and the yellow and the unknown. Now, you may ask, why would you find one of the control healthy individuals from Calcutta to be similar to the Western? And I would give two possible explanations. One is that these would be individuals who had been living in the U.S. for a number of years and then visited at the time and providing a sample as a control contribution because of their housemate or relative being ill and it's being useful for them to do. And the other is that it may very well could reflect the standard of living, and so those individuals may have a diet that's more like a Western diet, even though living in Calcutta. So I present in any case the conclusion that we can determine the Western diet from the Indian Calcutta diet very, very metagenomes very accurately. The other thing that we can do with this analysis is to provide an understanding of the antibiotic resistance factors present in the samples. So here, pooling the samples from the individuals for which the Calcutta lab could not identify a pathogen causative agent, and those that they could identify a pathogen agent, and then the healthy. You notice that it's a little disconcerting that tetracycline and the beta-lactamases are very, very common but also in the unknown etiology, a variety of other aminoglycosides and other trimethoprim resistance and so forth genes are present. Probably, my guess would be that that's because they're carrying pathogens with those antibiotic resistances. In any case, this is another demonstration of the power of the algorithms that we have been able to determine. And here is another aspect of being able to link, to collect the carbohydrate metabolism genes and to show that in this Vibrio cholerae, that there's a higher level of carbohydrate metabolism, and again, maybe a function of the diet of the individuals from whom the samples were collected. Now I'm going to move on to show how not only have we been able to, within minutes, literally minutes, 10 minutes, 5 minutes, once we have the shotgun sequence, to identify down to species and strain, bacteria, viruses, fungus, parasites, but also functional genes. Now we have teamed up with Qiagen, but their bioinformatics are even more powerful in terms of functional analysis. And so I'm going to ask Arnie to very quickly describe the method that's used in algorithms that's linked with our algorithms.
This one? I think this is better. What you'll see here is what we refer to as a workflow in the CLC genomics workbench on the left side. This is like a simple making a flowchart in PowerPoint. You can drag and drop tools in our toolbox into a flowchart and create an analysis workflow. We also provide these pre-configured but fully customizable to users to really get started fast. And this particular workflow here is configured to carry out functional profiling of microbial communities or the whole shotgun metagenomic data. And we begin with metagenome de novo assembly. This is the first step, which is very fast. As you see here, it takes between 2 to 27 minutes. There's sort of a linear relationship between the time and the number of reads that you submit. The next step is gene finding. We use the meta-gene mark algorithm to predict the gene models. This takes less than a second. Then the next stage is we annotate the gene models with function using, for example, information from gene ontology or protein family databases. Carry out a mapping step to really quantify the abundance of each gene functions by measuring the number of reads that map to each function and build functional profiles. All this together took on this particular machine that the analysis was executed on about 76 minutes on average per metagenome. But we've also tested this on this laptop that I'm giving you the presentation from, and it took a little bit less than 25 minutes for 5 million reads and still under 2 hours for 50 million reads. And that was the Yes, okay, good. I'll do that. The next stage then that we carried out here is we used, after having established all the functional profiles for all the metagenomes, we've carried out differential abundance analysis between the different groups, the healthy and the known and unknown etiologies. The idea here being is that we identify genes or gene functions that are both drastically overabundant in the groups of interest, yet for which you have a statistical significance. So what you see in those volcano plots here is on the x-axis differential abundance, so overabundance on the right side, for instance. And on the y-axis we see p-values or statistical significance, which really is a measure for the confidence that you have that this abundance fall is not random, that this is not a random observation. And there's various ways then from that to select the genes. One is to simply drag your mouse around the genes of interest, so above a certain confidence and above a certain differential in terms of abundance. Those are the red dots, and automatically the list of genes are then, or functions are then selected and you can submit them to further investigation.
So this allows us, with the basis of the p-value and fold abundance differential, to be able to characterize even further the gene capability. So we notice that in the healthy versus the unknown etiology, we find that in the unknown etiology, a much higher cytohemolysis capability. And again, in the using the protein analysis, we can also determine the presence of hemolysis, gene functions, and as well as the membrane-associated factors. The hemolysin really it confirms the fact that we've been able to isolate, for example, Salmonella, Shigella, and the enteropathogenic E. coli. So it is a mechanism for confirming the functionality of what we've been able to identify through the algorithms that we have built for rapid identification down to species and strain. So this gives us then the ability to determine the presence of gene functions, again, cytohemolysis in the case of healthy versus the known identification, and we find an even more powerful set of genes in the known, easily identified, isolated, and identified pathogens. And again, using the protein families approach, we get a confirmation of the hemolytic functions and the membrane and enterotoxin release that we, as a function of those microorganisms that we have identified. So, to summarize, we've been able to show significantly increased abundance in both the known and unknown pathogens that we have been able to identify using metagenomics approach, and the capabilities of those pathogens in cell killing and morphological modification. So the genes of these factors, these expressions, are really important and identified compared to the healthy or the asymptomatic isolates. And finally, one thing that I think is really, really fascinating as well, this is these are healthy, and this plot are the patients who, over a period of time, show a very decreased diversity. In fact, major, it's a loss of diversity and a selection for how should I say, the success of the pathogen in the later stages of the diarrheal disease process. The analysis of antimicrobial resistance is also very important, using the assembled metagenomes from the Research Finder database. And we find that the healthy samples, the NIH Human Microbiome Database, there's no resistance against these important antibiotics. Only a few of the NIH database samples have resistance against these, and on the other hand, the Indian samples show no resistance against this particular antibiotic, but a clear difference in the resistance patterns between the healthy and asymptomatic samples. So we're able to characterize then the populations according to the genes they carry for antibiotic resistance. So what I've shown you in a very short time, which represents many years' work, the capability now to do in about an hour and a half, calculated, combination of our approach coupled with a Qiagen approach that now is a single system that allows us to identify down to species and strain, to identify proportionality within a sample, to identify genes that are present for antibiotic resistance and for pathogenicity traits, and then to be able to characterize functionality in a very, very detailed way. And we now know that the microbial communities in the healthy volunteers suggest that the microbiome of healthy humans of Indian descent is very different from Western European. And this is persistent and determinable. The Indian population carries a low number, but much larger. We Westerners do carry very small numbers of pathogens as well, but the Indian population larger proportionality number of pathogenic microorganisms. Multiple pathogens readily identified from diseased patients. Very, very important point. Polymicrobial infections, more and more, I think, we'll be able to recognize, characterize, and be able to understand more effectively as the mechanism of the disease state. And then the metadata show that patients with profound watery diarrhea that were characterized and brought to the cholera hospital as cholera patients, in fact, carried primarily the E. coli complex, pathogenic E. coli, and Shigella species. Again, another important factor, which means that we have to redefine what is cholera. And then we also I would like to point out that the microbial community of the Indian population encodes really what might be termed an alarming rate of antibiotic resistance genes. And functional analysis of the Indian microbiome indicates a predominance of carbohydrate metabolism genes related to diet and an overabundance of the cytohemolysis genes that we observe in the unknown etiology that can probably help explain those factors that cause that particular disease state. We have a lot of pilot studies underway with top research hospitals in the U.S., in Europe, including Sweden, and in Amsterdam, and in the UK. We've been studying wound surgical infections, usually from tissues, aspirates, and swabs, orthopedic infections, neonatal sepsis, and so forth. And we're actively involved in studies of prosthetic joint infection as well as these others that are listed on the slide. So we're in the process of doing a lot of pilot studies and actual diagnostic for individual patients as well. The important point to make in closing is that I've been describing the work done in Calcutta, clinical application, but I would really like to emphasize that this has pharmaceutical application as well as food safety. In fact, we're doing studies right now with the FDA on identifying food pathogens. And we're doing a lot of work with water systems, both in Saudi Arabia as well as in Orange County and in California, to ensure that the reused water, sewage water that is drained now into the drinking water purification system does provide pathogen-free output that is safe for human consumption. The end-to-end, that is to take from a sample the extracted DNA sequence, and then from that sequence, within an hour and a half, to do all that I've described this morning. To do just the analysis for identification is probably less than 10 minutes. And then with the very elegant assembly and functional analysis, within an hour and a half, the total end-to-end provides us with a maximum amount of information. And you can see the very happy collaboration that Qiagen undertaken. So I thank you very, very much for coming in from what's a very busy, complicated meeting to listen, and I hope that you find this as exciting as I do, in that we really, I think all of us in this room and at this meeting are in the midst of a wonderful revolutionary time. We're modernizing microbiology. I'm married to a physicist, and he says, you know, if a physicist in 1890 would be alive today and walk into a physics lab, he or she, well, mostly he at that time, wouldn't be able to do a thing. But if Louis Pasteur walked into a microbiology lab, he'd just roll up his sleeves and say, give me a petri dish and an inoculating loop. We're changing that. We are now turning microbiology into a 21st century exciting. Just thank you very much.